All right, hello, welcome back. So now we're just going to go through and show a little bit more of what I was talking about in that table. This is all basically breaking down the table and showing specific cases where these things apply. Here in the yellow column, you have all kinds of diseases, and look at them all. Oh my gosh. And then you have organisms that are associated with this. With some of these diseases, it is one organism that causes it. With other diseases, potentially you can get a number of different bacteria that would cause a similar enough disease that it's just harder to tell. But each of these organisms is pathogenic because it contains a toxin. And a toxin is just a gene that can be expressed that gives the bacterium some kind of competitive advantage. And for the most part, those competitive advantages are normally used in fights with other bacteria. Because you have to imagine that to bacteria, we basically seem like a big shambling pile of slowly rotting meat. And so we are food that is there to be eaten. And what you find is that bacteria will beat each other up and make all kinds of wonderful antibiotics to kill off the competition so that they can be there at the feast and, yeah, basically have the biggest plate at the buffet line. However, there are a bunch of other bacteria that were normally fighting them, and so it's not that one wins, it's just that you have a sort of a permanent state of war. Very, very 1984, yeah. However, it also happens this way in nature. So, whereas something like a human body is a particularly high energy food source, and remembering that we're all dead on the outside, there's a lot to be eaten without us even knowing. In something like the soil, there's very low relative amount of carbon and relative amounts of other nutrients. And so, in order to survive, all these different bacteria that you would find out there need to be very, very creative in how they are scavenging nutrients and in how they are essentially poisoning and murdering the competition. So, that's where toxins come from. I think, interestingly, at least for me, some of these toxins, like the cholera toxin, are transmitted on viruses. So, the virus infects the bacterium and then it delivers some genes to the bacterium, which includes a toxin gene. If the bacterium doesn't die, then the virus also gets to survive because it becomes part of the DNA of that bacterium, and the bacterium gets a little boost, like a little buff, because it's now carrying a toxin that makes it more toxic to its potential foes. And so you'll see that the most sort of devastating Vibrio strains are very similar to E. coli, but with multiple copies of these cholera toxin containing bacteriophage, like bacterial viruses, inserted there. Yeah, it's just they're basically loading up their host with armaments in exchange for compromising the DNA of that host. So, interesting, I think. Yeah, these are some of the effects of those toxins, and they're great. You don't need to memorize these. You may need to memorize them for your medical microbiology class, but not for here. This is just showing that various bad things can happen when these toxins are produced, and these are bad things that happen to the human host. So, what do we do about this? Obviously, as you know, shambling piles of meat, we don't want to be eaten by bacteria, and that's mainly what our immune system is there for, to keep these little buggers off. So, ordinarily, you would try to make antibodies. And so the way a toxin would work is that the toxins will bind to cellular receptors. And when they are pulled into the cell, they'll then be released and they'll do different things to the cell. Each one of these works in a slightly different way, but they're going to disrupt the physiology of the cell. They may even kill the cell, like this one's gone gray and kind of a notched nucleus to show that it's probably necrotic. Um, and sometimes there are also toxin complexes where the toxin, you find this with like botulinum toxin has several different components. A lot of these toxins are multi-component. One component will bind to the receptor, and the other component will then be released from the receptor by little proteases. and then can penetrate into the cell and do its damage. So the point here is that if you make antibodies against the toxin, you can actually prevent the toxin from binding, and you want to really block the part that would bind to the cell, because if that doesn't bind, then the little red part, the effector part, never is going to get released, and so you're not going to have a problem, basically. So that is neutralization at its most simple. It's just sticking to the thing that's going to try to stick to you, and basically getting there first. So when we vaccinate, for example, against really bad diseases, like you can vaccinate against anthrax, we try to make antibodies against the toxins quite often. And some of the childhood vaccines you will have taken are toxin vaccines, like vaccines against the toxin. They're not giving you toxin, they're immunizing you against toxin, really. All right. This also applies, though, to larger things. So bigger than a toxin, like a toxin is usually just a protein, you would have something like a virus, and they're a little bigger than a toxin. So viruses are going to try to do the same thing. They're going to try to first bind to the cell, and then a virus isn't going to be able to actually do anything unless it gets into the cytoplasm of a cell. And then once the virus, which is really just this little sequence, this little chain of either RNA or DNA, once that gets into the cell is infected, and that cell is probably going to die sooner or later, and it's better for the body if it's sooner. So what do? We basically have to stop this before it happens, because the only time when antibodies can get a fair shot at the virus is when it's outside of the cell, before it has started the infection. Once the virus attaches to the cell and is pulled in a little endosome, and almost all of them do this, it'll be released, and at this point the proteins aren't even attached to the real troublemaker, which is the genome inside, and it's actually the genome that's going to take over the cell. It's really the information of a virus that causes the damage, and also encodes all these little proteins that make up the nice little red shell. So yeah, blocking a virus before it can enter, that's good enough to prevent an entry event. Yes. So dumb you almost don't even need to say it, right? That's what neutralization means, and that's one of the things that antibodies are very good at. So think which antibodies are going to work where. If we're talking about something like your mucosal membranes, really the only ones that get to those mucosal membranes in large enough quantities are going to be the dimeric IgA molecules. And so if you get neutralization at those membranes, it's going to be dimeric IgA that's doing the heavy lifting for you. If the virus gets through that barrier and gets into some cells, it then may have access to the bloodstream. And that's fine. If it's in tissues or in the bloodstream, you've got IgG that's going to be there and is going to potentially be able to stop the virus before it can spread a little bit more. And you would also make a little IgD and IgE and monomeric IgA that would help a little bit. But it's mostly those two that are going to do the bulk of the work, IgG and IgA. IgA on the outside, IgG on the inside. All right. You're probably asking yourself, does this work with bacteria? And of course it works with bacteria. The only problem with this is that the number of antibodies that you need goes up as the size goes up. The number of antibodies actually scales with the surface area, so it's like the square of the diameter, I guess. Is that right? Yeah, I think that's right. You may only take a couple of antibodies to knock out a virus, and you may take hundreds of antibodies to knock out a bacterium, and there are some wonderful papers where people have actually tested this out. And they've shown that little viruses can be knocked out by as few as 4 antibodies per virus, and big viruses, you may need 100 antibodies per virus, and things like bacteria, you may need hundreds to even thousands of antibodies to knock out a single bacterium. Because remember, you got to cover all the parts of the bacteria that would be binding to cells, and then once they bind to cells, they would then get pulled into cells, and once the bacterium is inside the cell, it has access to the cytoplasm, which is where all the nice proteins and ATP and all that good stuff are located. And that's what bacteria really want. Cells have that membrane on the outside that makes it a little bit difficult for the bacteria to find anything to eat. And so they just attach themselves to the window like flies in summer and basically see if they can get pulled inside, trick their way inside. And so antibodies can stop these, but the number of antibodies that you need to be able to block bacteria from infecting is very high. So quite often you'll have a difficult time fighting off bacterial infections at the start. But from the sort of mid-infection to late infection, as you're cleaning up the infection, that's when you'll have the highest antibody levels, and that's when the antibodies can actually do the most good, because they're going to be working in conjunction with all of your T cells, for example, and all of the other components of your immune system. So antibodies alone are okay at preventing entry and so-so at doing cleanup, but they work best when they are supported by the rest of a functional immune system. All right, last little bit in this chapter is talking about antigen-antibody complexes. So imagine this great big planet here is like a bacterium surface, and imagine you made an IgM that binds to something on there. This little IgM can then sit down and it can interact with several copies of the same antigen. If it's something like a cell wall protein or something like that, extracellular membrane receptor. And then remember, C1q needs several antibodies, FCs, to be in the same region in order to bind, and IgM comes with that already pre-built. It's baked right in. And so one IgM can bind to a C1q and can trigger the first step, and you'll start activating. And so, yeah, C1q is going to activate C1R, which remember C1R and C1S are these little proteases up here in purple and green for whatever reason. And those are then going to go and activate some of the other complement members in the classical complement activation cascade. The other way to do it is going to be with IgG, and so IgG is going to try and bind to those same receptors if it can, which is great. And then if you happen to have two IgGs that are bound to their receptors that are close enough that their FCs are within sort of grabbing distance for a C1q, then you can potentially activate C1R, which will then activate C1s, and then QRS, and you are off to the races. You can start building up those pre-activation complexes and eventually get to the big old C5 activation complex. And from there, there's pretty much no turning back once that thing's turned on. So here are electron microscope pictures showing the same thing. And so this is actually IgM, and this is one IgM molecule. And so you can see it's all flat, and each of these lumps up here is a one of the FABs, essentially. And so they're all chained together down here in the center. And it's a little wobblier than it looks when we draw it, but it's basically the same thing. And here are IgMs that are sort of folded. So these are folded so that some of the domains are pointing down, and the center region where all the FCs come together is flexed up. And this is going to make a very inviting binding site for something like C1q. Okay, and I believe this is the last component in this chapter, section 10-20. So the other thing that your antibodies can do that helps sort of keep you safe is that antibodies can form these complexes. So if you get a multimeric antigen, so like say an antigen is made of 5 copies of the same thing, which is what they're showing here, which something like a virus is going to be coded in tens to hundreds of copies of the same protein. So it's something that works well for viruses. The same thing with some toxins. There'll be multimers like that. And so you can get multiple antibodies chaining multiple antigens. The antigens are the red sort of flower-shaped things here at the top. And this is an antigen-antibody complex. And so once you have those antigen-antibody complexes, that is going to potentially lead to deposition of some C3bs. And yeah, once you have C3bs there, you can then bring in potentially C1qs will attach onto the FC regions. And you can also potentially get a C3 convertase there as well. And between those will give you multiple ways of activating the complement pathway. So basically, if you have enough antigen floating around and enough antibodies against the antigen to chain all this together in a big biological gloop, then that will activate the complement cascade, and basically this is a thing that needs to be either gobbled up or just destroyed, blown up. Yeah. All right. So what this can do is that there is a complement receptor called CR1 on red blood cells, and it'll bind to these complexes that have already bound C3b on there. And if it's holding on to a C3b that's holding on to an antigen that's holding on to an antibody, then potentially, yeah, you've got a way to pick up a whole great big complex. It's just sort of the way you'd pick up, I don't know, 6 bottles of Dr. Pepper. They got a little ring there to hold them all together so that you just grab one and all the other 5 come with them. Yeah, that analogy was brought to you by the Dr. Pepper Corporation of somewhere, Texas. Enjoy responsibly. All right, in the spleen and liver, phagocytic cells remove the immune complexes from the erythrocyte surface. So you've got all these white blood cells. In particular, neutrophils are going to be good at this, but also macrophages. And they're going to scan these red blood cells, and if they see a whole bunch of just immuno nonsense stuck to the outside of there, including C3bs attached to antigens, attached to antibodies, attached to whatever, then they will be able to scavenge these. So they'll have both complement receptors that are able to bind to that C3b, and they will also have FC receptors that are able to bind to antibodies. And if they get sort of a nice big chunk with several of these together, then they will sort of swallow that up, bring it in, and break it down. And the good thing about these phagocytic cells is that when they do this, they will then generally go on to display at least a little bit of whatever it is that they have found. At least the professional antigen-presenting cells, like macrophages and dendritic cells, are going to be very good at this. Yeah, neutrophils, they do their best. They're not smart, but they are effective. So yeah, don't count them out anyway. All right, and the very, very last thing is, how do we end up with secretory IgA on the mucosal immune system? And so this is like an expanded version of what we were talking about with that earlier picture. So here's our polymeric immunoglobulin receptor, and IgA is going to be held together. You're going to have 2 of these IgA molecules held together by what they call the J chain, which looks like this little I don't know if I said croquet wicket, would that make sense? It looks like the letter U. Here it's upside down. And that's going to hold those together, and then they're also going to interact with the immunoglobulin FC receptor, which is requiring binding from 2 of these at the same time. So it's going to tend to pick up these dimers that are held together with the J chain at just the right distance. And that's going to get transported up here, avoiding the lysosome to the gut lumen. And then when it's there, actually are going to release this by cleaving the receptor, and launching a complex is going to have your IgA dimer, and there's also going to be a secretory component. They can then fall off or stay there. It doesn't really affect the function of anything from that point, as far as I'm aware. So that's how it happens. That is how the sausage is made, and yeah, it's a good thing. It's a good thing too. So, all right. And then I just wanted to put this in there because I like it. So once the soluble secretory IgA, they'll sometimes show that as little s, capital I, little g, capital A. When that is out there, it can then do the normal antibody things. It can bind toxins before they're able to get into the cell and do bad things, and it can bind to bacteria. And once it is bound to these things like toxins and bacteria, they can also get transported back down. So if there's another little transporter there, such as dectin-1, it can grab onto an antibody that's found something. It's basically just like fishing, except that you just launch a bunch of fish hooks and then reel in the fish hooks that happen to have caught something. So dectin-1 is able to grab onto these antibody-antigen complexes, and it'll transport them back down, and then it'll flip them over and feed them to a protein called DC sign, which is on dendritic cells. That's what the DC part stands for, and I think this was one of the earliest markers that people knew. So they sort of crammed the acronym in so that this sign of a cell being a dendritic cell could be called a DC sign. Yeah. Anyway, once a dendritic cell picks this up and it says, "Ooh, our IgAs have caught something," it can release some other signaling molecules. In this case, it's going to release interleukin number 10, and that is then going to go on and have downstream signaling consequences on various type of cells. And you could go into those. There's probably a chapter coming up in the book where they go into all the things that IL-10 does and doesn't do. But for now, just know that a message has been sent in response to this. And I think that's going to be good enough for this section. All right, so hope that all made sense, and thanks very much.